Okay, so section 3 is talking about peptide bond formation. Amino acids are connected to one another by peptide bonds, and which connects the C terminus to the N terminus. Peptides are basically just amino acids connected to each other. And we have different types of peptides. We have polypeptides and oligopeptides. So oligopeptides are peptides that have less than 20 amino acids in them. Polypeptides, on the other hand, is the peptides that have more than 20 amino acids. Residues are basically just amino acids. You might hear residues, amino acids, or even amino acid residues. They just mean the same thing. And then we have dipeptides and tripeptides, which means those are made out of 2 and 3 amino acids. So formation of a peptide bond actually results in an extra water molecule. So you gain a water molecule when a peptide bond forms. And if you want to remove that peptide bond, you're going to need a water molecule. So section 4 of chapter 1 is talking about primary and secondary protein structure. So primary structure is the linear arrangement of amino acids coded in an organism's DNA. So basically just amino acids in a chain bonded together with peptide bonds. That's it. Very simple. The primary structure of a protein can be determined by a laboratory technique called sequencing. Again, if you do take genetics, you will learn about this a lot more. After hydrogen bonding between the nearby amino acids happen, that results in secondary protein structure. There are two most common secondary structures, which are alpha helix and beta plated sheets. We have two types of beta plated sheets, which are parallel and antiparallel. For the alpha helix, hydrogen bonding happens between every fourth amino acid. So to be more specific, the hydrogen bonding happens between a carbonyl oxygen atom and an amide hydrogen atom, fourth residues down the chain. As we discussed earlier, proline is a special amino acid because the amino group is actually in the R group. So basically the nitrogen of the amino group is located inside the variant or the R group. That is useful in the secondary structure because of proline's rigid cyclic structure. That can cause and introduce kinks in the alpha helix or create turns in the beta plated sheets. Proline is commonly found in the first residue of an alpha helix, and also it is in the edge strands of the beta plated sheets. Section 5 talks about tertiary and quaternary protein structure. So in general, we have two types of protein structure. One is globular and the second one is fibrous. Globular tends to be spherical, and fibrous have structure that resembles sheets or long strands. So the tertiary structures are mostly determined by hydrophilic or hydrophobic interaction between the R groups of the amino acids. So, for example, if we already discussed this in section 1, but if amino acid is hydrophilic, it will be on the outer side of the protein, and if it's hydrophobic, it will be in the interior or in the inner part of the protein. Okay, so molten globule. So if we were to go from tertiary structure all the way to primary structure, we would have to go through steps. And basically the intermediate state in the folding of a protein is called molten globule. So as you can see here, this is the unfolded part, and as we go down, we got the molten globule, and then we have the native state, which is the tertiary protein structure. Quaternary structures contain 2 or more polypeptide chains, and each of the polypeptide chains are called a subunit. The roles of quaternary structures are that they can be more stable by reducing the surface area of the complex. They can reduce the amount of DNA needed to encode the protein complex, which is very important for viruses. They can bring catalytic sites closer together, and also they can induce allosteric effects. Prosthetic groups are non-amino acid component that is covalently attached to the conjugated proteins. And the conjugated proteins get part of their function from the prosthetic groups. So for example, I have a hemoglobin molecule right here, which has 4 subunits. And each of the 4 subunits have a prosthetic group that is called heme. And in the core of heme, there is an iron molecule, which, as we all know, carries oxygen. So basically, one hemoglobin molecule can carry 4 oxygen molecules. Now, there are some naming involved with them. If the prosthetic groups are lipids, the protein is going to be called lipoproteins. Carbohydrates would be glycoproteins. Nucleic acid would be nucleoproteins. Section 6 talks about denaturation. So what causes denaturation of the proteins are heat and solutes. So how can heat denature a protein? When the temperature of a protein increases, its average kinetic energy increases. And that extra energy at high temperatures is enough to overcome the hydrophobic interactions that hold the protein together. And so basically, the proteins that are in the core of the protein, the amino acids in the core of the protein, which are hydrophobic, they won't keep the tertiary structures anymore because the kinetic energy is very high. So we had the figure right here. You see, as the energy increases, it goes from native state to getting unfolded, and it goes through the molten globule, of course, which is the intermediate state. The second cause of denaturation are solutes. The book specifically talks about urea, which breaks disulfide bridges. The book also gives an example about denaturation of proteins, which is when you cook an egg, when the egg turns from being clear to white, you are basically denaturing the proteins in the egg. And fun fact, it is possible to actually unboil an egg using urea with centrifugation. I will put the video link for that in the description, so if you're interested, it's pretty cool. You can just watch it. It's a few minutes long. And that brings us to the end. That was chapter 1 of Kaplan Biochemistry. So let me know if you have any questions. Thank you for watching, and if you can, please like and subscribe.